My pleasure to introduce Professor Stephen Holdcroft, who is a WIN seminar speaker today. Professor Holdcroft was born in the UK and he received his bachelor's degree in chemistry from the University of Salford and his PhD in 1983 from Simon Fraser University under the supervision of Professor Emeritus Lionel Fund. He was a postdoctoral and NSERC postdoctoral fellow at the University of Toronto with Professor James Gillette after that, and returned to Simon Fraser in 1990 to accept a faculty position in the Department of Chemistry, and he became a full professor in 1998. Steve has been on half-time secondment to the Institute of Fuel Cell Innovation of the National Research Council of Canada in Vancouver, assisting in the development and implementation of a strategic plan of research for NRC's National Fuel Cell Research Program. And at NRC, he's been a group leader and has led numerous fuel cell research programs. His interests span materials chemistry, macromolecular science, and electrochemistry, with a focus on electrically conductive and ionically conductive polymers for plastic electronics and for fuel cells. He's the author or co-author of more than 100 refereed journal articles and 3 book chapters, and he also holds several patents and sits on a number of international and national advisory panels. He's a fellow of the Chemical Institute of Canada and a keen supporter of Newcastle United.
Newcastle United? I don't think so. Try the other one.
So please join me in welcoming Steve and his talk, Sleeping Policemen on the Hydrogen Highway, Addressing Challenges in PEM Fuel Cell Technology Through Basic Research. Welcome.
Thank you very much. It's a great pleasure to come to your university. I was dropped off this morning at the new building, not knowing that there was nobody in it. So it was very nice and spacious, and I'm sure it's going to be wonderful when you all move in. A few years ago, I would have given a lecture, and I would have entitled it Roadblocks in the Hydrogen Highway. But progress has been so fast and so rapid recently that now we only have little bumps along the road, and that it's almost inevitable that there will be a fuel cell technology. Now, if you're coming from the east, you might not realize that when I'm talking about sleeping policemen, I'm talking about these little bumps in the road that slow you down but don't prevent you. I'm not talking about this type of person. And they really are called sleeping policemen. We don't just use this in a slang way. You can see signs for that. I'm coming from Simon Fraser University. It's on the border between Vancouver and Burnaby. And it's looking north here towards Garibaldi Provincial Park. And it's not a bad place to be. If you look west, you can see downtown Vancouver. You can see Stanley Park. Oh, this one's a little bit better of a color, I think. Stanley Park around here. And just on the grounds of this other university, name escapes me right now, is the National Research Council. And that's where I've been half-time for over 12 years now. And this was intended to be a center for development of fuel cell technology to support local industry. And so just down the road, actually in Burnaby, is a company called Ballard Power Systems, another company called Automotive Fuel Cell Corporation, Collaboration Corporation, which is largely owned by Daimler, but also owned by Ford and Ballard. And then right next to it is a new plant, a new facility designed by Mercedes-Benz Canada. And that plant opened just recently. 2 years ago, there was an announcement that Mercedes-Benz would invest 50 million euros to build a manufacturing plant for their F-cell vehicles to make fuel cell stacks. This is really quite remarkable because now you've got a German company deciding to come and place their first facility for manufacturing fuel cells in one of the most expensive places in the world. They could have gone anywhere. And they do that because locally in Vancouver, you have all this knowledge of fuel cell technology. We had the Institute of Fuel Cell Innovation, which Dr. Carty spearheaded and was his brainchild, specifically designed to form a fuel cell cluster in Canada. If you're ever interested what a manufacturing plant looks like for fuel cells, it might be very different than what you expect for a normal automotive. It's almost like a Class 100 clean room. They don't make a lot, it makes 5 stacks a day. That's about all the companies do. There's some limitations. But from a Canadian perspective, what we need to do and what they need to do is reduce the costs of fuel cells in order that they will maintain their presence in Canada. They're not the only company working in fuel cells in Canada. They're spread across west coast to east coast. There's a company we work with in Ontario, and I got this news alert just 2-3 days ago about some new opportunity in fuel cells. And I draw your attention because there's a really unfortunately placed pole here, which gives another meaning to biomass energy, but that's another story. So just in case you've been kept in the dark for a long time on what a fuel cell is, the recent embodiment that people are excited about involves a plastic material that conducts protons and separates 2 electrodes. At one of them, one oxidizes hydrogen to protons. At the other one reduces oxygen to water. The protons go from anode to cathode. So there's a couple of integral materials here. One is the membrane that transports protons, and the other one is the catalyst. Now, if you take a sheet like this and you make a very thin, what we call the membrane electrode assembly of the membrane, the catalyst, you bring on the flow fields to bring hydrogen and oxygen in. You put that together, a sheet size would generate about 240 watts of power. 240 watts, what's that? Well, that's about 4 light bulbs. So it's not very much power. You can't really do a great deal with that in terms of large-scale energy applications. For an automotive application, we need 80,000 watts. 80,000 watts. So now you realize you have to make stacks of these in series that are hundreds of components. And when you take into account you need plates to bring in flow channels, sealants, gaskets, you're looking at 1,000 components to make a single stack. One can do that, and you place them in nice little containers. This one from Nissan. You can go to Toyota, Honda, Daimler. They all have their versions. And that's just one of the components in the automotive powertrain. You have the stack. You have a balance of plant which controls humidity and temperature. You need to store the hydrogen. You still need batteries. And then you have converters, radiators, and so on. Now, there have been great advances in fuel cell technology in the last 10 years. If you go back in around 2002, there was a projected cost of almost $300 per kilowatt for a fuel cell stack. That has dropped significantly. Today, it's just under $50 per kilowatt. And one has to bring this down to about $30 a kilowatt. Why 30? Well, we know that the cost of an engine, an internal combustion engine, is around $2,500, $3,000. I know this very intimately because I drive standard, and I blew my car up about a year ago, and I had to get a new engine. and it cost me about $3,000. So this is the target. You multiply this by 80,000, then you get an idea of the cost. And that cost involves that stack that's made, and also the balance of plant, the process to keep the fuel cell humidified and at the right temperature. So without going into a great deal of schematics, if you look at the flow diagram for what's required in a fuel cell, and you look at the hydrogen storage, and you look at the fuel cell stack, and you look at all the balance of plant to keep this stack happy and moist, and you project the cost associated with making, say, 100,000 of these, like a typical Toyota Corolla, then you realize the cost reductions need to come down in two components. One is stack, and also the balance of plant. And most chemists focus on this, largely because when you do a cost analysis on mass production, everything comes down with economy of scales, except the platinum. It's a commodity, and so you quickly figure out that the platinum will be 50% the cost of a fuel cell stack. So this is one area that needs significantly addressing to reduce the cost of fuel cells. Many people are involved in this worldwide. We're involved in this. We picked off where Jianghu left off and trying to build a network. We're successful in gaining a kind of an NSERC network for lowering platinum costs for fuel cells. It's a 5-year program. It goes from coast to coast with researchers all across the country. Not all researchers across the country, but several of them are located here. And it really is a challenge in nanotechnology, because the group as a whole is looking at nanofibers, graphene, nano-controlled catalysts of different composition, and it's a real complement of people, and we have a few themes that I won't go through. Now, to get down to $30 a kilowatt, it's assumed you have to reduce the platinum. But it's also assumed that you can get rid of most of the balance of plant. And this is used to keep this stack at a relatively constant humidity and constant temperature. And there's a big assumption that what the automotive designers make is that in 5-10 years you can remove this. But is it possible? And this is where the science and chemistry can make a very big impact. So why is humidity important? Well, if you look at the archetypal membrane in a fuel cell, it's based on a Teflon backbone with a triflic acid side group. They phase separate into ionic domains and fluorous domains. And even if you expose this to the open atmosphere, these domains are so hygroscopic that they absorb moisture, they dissociate the protons, and these protons are now able to be highly mobile. The water is also mobile, and that's absolutely critical, but I won't talk about that. When it comes to the water transport, there's generally, and simplifying this, two types. There's water which is a proton hopping from one water molecule to another. We call that the Grotius mechanism. But there's also the other way, and that's the vehicular. That's like a movement of hydronium ions, just like a normal cation would move. And it's the fact of this hydrogen proton hopping that gives the very high rates of diffusion and conductivity in acid media, unlike sodium, lithium ions, and so on. However, if you look at water, and that proton hopping mechanism needs liquid water. And so if you look at a hydrated membrane, such as Nafion, and you can analyze for the type of water spectroscopically or by DSC, you find is generally two classes. Again, a simplification. You have half of it is basically like bulk water, it's freezable, and the other half is strongly bound, highly polarized. And when the relative humidity is lowered or the temperature is raised, the membrane loses water. And it's the liquid water that is lost. And this is the water that gives the very high conductivity for the protons. And so as you drop the relative humidity, the proton conductivity for this membrane drops very quickly. And that gives a very high resistance, means a loss of cell voltage, means hot spots where you can get degradation. And so we would like this as flat as possible. And so in this field, there's a huge amount of research trying to replace these perfluorinated ionomers. And there's only like 5 companies in the world that make these because the basic component, TFE, is a controlled substance. We can't get it in our lab. You can't transport it. And so one would like to move to hydrocarbon materials or semi-fluorous materials. One, because these are very expensive, but also what people don't realize, they have very high gas permeability. The perfluorinated, very high gas permeability. And you may ask yourself, well, what's the big deal with that? Well, when you have high gas permeability, it means hydrogen can transfer to the cathode, and it means oxygen can transfer to the anode. And when that happens, then you can get the possibility of the generation of a variety of radicals. And this has a big implication on degradation pathways in fuel cells. Because now we're at the point of commercialization. Manufacturers are starting to look at how long these live. They want them to live for 20,000 hours. And so now durability becomes an issue. And so these radicals, they thin the membranes, they puncture the membranes, and that basically kills the cell. And if the cell gets killed, the whole stack gets killed. And that's a $3,000 problem. And so they want materials that are less permeable. And so for that reason, there's a great deal of interest looking at developing new types of hydrocarbon membranes that can replace perfluorosulfonic acids. So how easy is that? Well, you can go to, you can grab any type of polymer. You might be able to sulfonate it and put on different amounts of acid groups. And the parameter that we use is the ion exchange capacity. This is basically the number of sulfonic acids you have per gram of material. And so for a variety of materials, you can increase the ion exchange capacity. You have more protons, you get higher conductivity. But they have these unusual properties of increasing conductivity and then decreasing. And they happen at different ion exchange capacities. And so this is something that we started to look at 15, 20 years ago with a view to understanding and then developing new materials. So before I go into that, let me just tell you a few of the parameters I'll measure, and I'll keep them as simple as possible because it's a relatively broad, a group with broad expertise. When you look at proton conductivity through a material, it basically comes down to 2 factors: the mobility of that carrier and the number of those carriers. The number is associated with the proton concentration. The mobility is associated with the tortuosity or lack of tortuosity of the channel, but also the extent by which the proton is held to the anion. So if there's very little water around, probably the channel is very constricted, but if there's little water around also, the proton is very tightly bound to the anion. So in both those instances, the mobility will be very low. The other thing to realize is, as you increase the acid content of these materials, the water content increases disproportionately. So you've got an increasing osmotic pressure, but it can only be withheld by the elastic forces of the polymer. And so they're gaining more water than they really should. And so we measure this waters per sulfonic acid group, and they increase dramatically. So even though we're attaching more acid groups, the concentration of protons can actually be falling. Now, instead of plotting the ion exchange capacity, if we plot the water volume, water content of the membrane, as we increase the ion exchange capacity, the mobility of the proton rises. The more water that's present, the higher the mobility. But the acid concentration is dropping. And so somewhere along the line, you get a maximum, increasing proton conductivity, decreasing concentration. And so the differences in all these positions of maximum conductivity have got more to do with the way they absorb water and how they affect the concentration, in addition to the mobility. So a few years ago, we asked a question, if you could design from scratch and you had unlimited synthetic capabilities, what polymer would you design for a maximum proton conductivity for a fuel cell, for instance? Would it be a single anion at the end of a graft chain? Would you have multiple ions? Would you have random structures or block structures? At the time, no one really knew, and even today, we're only a few steps closer. So we tried to look at a systematic approach. We tried to look at two different systems where we could reasonably accurately control the morphology. One using a die block system where we know they should all to go into these different phases which are known for die block polymers. And another one from a graft system where you can have long grafts or short grafts. But the idea is to try to have very, very similar compositions, because if you change anything along the backbone, you've changed too many properties and the water absorption properties will change. You get different results. So it's trying to keep the materials as closely together compositionally as possible. With the added difficulty that to conduct protons, you need a lot of ions. And most systems that you put ions on will become surfactants. And so we were restricted in this work to having fluorous backbones, something that had very high cohesive energy density, so that they wouldn't dissolve. They would still remain as membranes. That's not so easy, because the methods used to polymerize fluorous materials are not generally compatible with polymerizing a second block. And so we designed a material using gaseous fluorous monomers with what's known as a chain transfer polymerization. We modified that, so we ended up with the hydrogen on one end and the chloro group on the other end. And we use a technique made famous by Matuszewski and Carnegie Mellon, which is called atom transfer radical polymerization. And these grow from chloro groups. And so we can control the length of this chain, and we did this using styrenic groups. And then eventually you can grow this a certain length, then sulfonate, and now you've got a die block with controlled lengths, controlled number of acids. It's pretty nasty chemistry, and not a lot of people would do this. These will never be useful in fuel cells. Because the styrene contains quite a labile hydrogen, easily abstracted. So they're model systems. We did have the idea of using trifluorostyrene. We spent a lot of work on that, and we were partially successful, but not enough to make us rich and so on. Now, we can take that analogy and replace one of the components with a chloro group, chloro-tetrafluoroethylene, and now we have a pendant chloro. We do the same chemistry, and we can grow a chain of fixed length and sulfonate. So two different types of structures. Now I'll start right from what could have been a conclusion, is that when you look at these two structures, and they're almost identical in composition, there's very few chloros, maybe 1 in 100. And you can see that the structures give very different images on a transition electron microscope. One system gives highly organized lamella, where these are stained. So this would represent the ionic groups, and the white is the fluorus. And if you look at the graft, you see these completely disordered structures with very, very tiny ionic aggregates and very tiny fluorus groups. When we got this, we published it in quite a high-impact journal. And it created a bit of a stir. We got a lot of input from industry, a lot of interest. Many groups started developing die block systems because these look like perfect channels for proton conduction. However, they didn't know what we knew at that time. And what we found out was these channels, although they look really useful to have in a fuel cell, have a few problems. And I'll have a few slides to show what those are. If you just compare these two structures, so almost same compositions, different architecture, different morphology, the block systems absorb a lot of water really quickly as you increase the acid content. So much so that after a value of about 1 millimole of sulfonic acid per gram of material, they essentially dissolve. And if you think about it, it makes sense, because the water will come into these hydrophilic ionic groups and swell, and there's nothing holding them together. So very quickly, they just fall apart and dissolve. Whereas these structures have a very intimately connected bicontinuous structure of fluorus and ionic domains, and you can really load up the acid content without them dissolving. And I'll show you why that's important later. The conductivities are similar, the die blocks, but they're shifted over. But these contain huge amounts of water. They're almost like gels, completely useless. Whereas these are much more like a normal polymer membrane, a piece of plastic. A few things about the die blocks, what we learned from them though. We can control the length of these blocks to different extents. We can sulfonate to different extents. A lot of characterization goes into that. And you can place them on a phase diagram known for block copolymers. And basically, this phase diagram plots on one axis the chemical dissimilarity between one of the blocks and the other. The chemical dissimilarity, so a big difference between ionic and fluorus, and the volume fraction of one of the blocks on the y-axis. And basically, it plots out, as you go across here, you go from spheres, cylinders, and various other transformation, then an inversion of structure as you go past the 50% of the blocks. And what we had done is we had made quite a few blocks. Most of them fit in the lamella, and 1 or 2 fit into what's known as a disordered structure, and that was these. So very short chains gave us very disordered, longer chains gave very ordered. And our perception was these should give us way higher conductivity. These are the systems we wanted, but I've kind of alluded to that that's not what happened. And if we look at conductivity, and I won't go through each of these individually, but take my word for it that it's actually the disordered structures that gave the highest conductivity, as opposed to the lamella structures. And the lamella structures suffered in another area, in that the lamella always went in plane with either the casting substrate or the extrusion dimension. Whereas in a fuel cell, you want protons going orthogonal, perpendicular. And most of our conductivity was going in this direction. So there are two major hits that we took from having truly organized structures. Now, we couldn't understand why these should be their conductivity was so low and we found out when we teamed up with physicists from our department, Barbara Friskin and the students, and we did some small angle neutron scattering on these, and from the scattering, which I won't explain in any detail, you can see the fluorus and the ionic domains quite clearly. But then when we looked into the ionic domains, we found extra scattering functions. This made perfect sense as soon as we saw it, because in this ionic domain is not pure iron, but it's really hydrocarbon and iron. And so we have tiny aggregates of ions within our ionic channel. And it turns out it's the percolation of the ionic aggregates within this channel that often dictates the mobility of the proton, not necessarily this gross organization that we've got. So this is a very, very important finding for us, and I think for the community. What we also found out is the more perfect the structure, obtained by going to longer and longer lengths, better lamella, then because of the density difference between the two chains, the degree of sulfonation of this block was actually lower the longer the styrene length. And because the degree of sulfonation was lower, there were fewer acid groups for a given ion exchange capacity. It meant the ionic aggregates in the channel were much further distributed. So we realized we want high concentrations of ions within our ionic region. So that allows me to move into the graphs, because that's what turns out we observed. In the graph materials, we can control the length of the side chain, and by the number of chloro groups, the distance between the chains. Again, keeping the composition similar or constant. A lot of work goes into the characterization from the chemical perspective, but just let me talk very briefly how we do a lot of our morphological characterizations. We use XRD to look at crystallization of the backbone, of the fluorous backbone, and determine the size of the crystallites. We use small angle X-ray scattering to look at the size of and scattering from the crystallites, and also scattering from our ionic aggregates. And we can differentiate crystalline regions from ionic regions using small angle neutron scattering, using phase contrast variation, where you can imbibe different amounts of D2O and water, so you can tune in and tune out the hydrophilic regions so it matches the phase of the fluorous region. So we can truly get a good idea of the morphology. So these particular structures are slightly different. The way we characterize them is that they're really based on the graft chain, which is in this case styrenic, with tiny small aggregates of ions. And these is all kept together by the backbone, which is fluorous, and parts of them crystallize. So without this fluorous backbone, this would just dissolve instantly. But it's the crystallites that are formed, and the connection of the crystallites that hold this thing together and allow it to absorb water, but not swell and dissolve. And so the kind of things we're interested in looking at the organization of these little white fluorous domains, whether it's a liquid-like order or whether it's much more systematic order. This is an extreme. And also now when we look at the TEMs of these structures, and by TEM you may think you've got an ionic aggregate, but really it's a really messy aggregate. It's got hydrocarbon, it's got ions, but from a TEM it just looks like ionic. It's just highly a lot of electron density scattering. So within what is viewed as a cluster of ions, we actually have very small clusters of ions, these red parts. There's about 10 ions per aggregate, and there are about 10 aggregates per little cluster. The rest is hydrocarbon. And so it's the connection of all these ions that give rise to a lot of connectivity, and then the ions, aggregates, then have to form connections. When you learn this, you can then start to design and tune the material. So the kind of information we get, the size of these crystallites that hold everything together are on the order of about 2 nanometers radius, and the volume fraction of about 20%. And if we look at the ionic aggregates, we can find they're about often between 2 and 5, 6 nanometers. And they're rich in ions, but not all the ions go inside the aggregates. Some of them can't find inside, and it's the ones that are on the chains that also absorb water. And so when they take up water, the aggregate swells, but then you start to form channels between the aggregates. And so the protons are moving within the aggregate and through the aggregates. And we're looking at dimensions on the order of 1, 2, 3 nanometers. That helps us understand how to design materials. Once you have an idea of the size of the domains, then you can do some simulation. And this is some work of my student's thesis and from Barbara's student's thesis. You can simulate the scattering from ionic clusters and the thickness of your ultramicrotome sample, and you can simulate what a TEM image would look like. And they look remarkably, well, you can make them to look remarkably like the TEMs you actually get. So that's the basis of the techniques. Let me say what you can do and how we've modified the chemistry. You can increase the length of the side chains, and you can determine the size of the clusters and how that correlates the aggregate size and the conductivities. And so just very quickly, as you grow the side chain length longer, then you get very different water absorption properties. These would be the long chains, medium chains, short chains, and the conductivities are quite different. And we can explain this now quite easily, that you can see if you make this chain longer, then you're getting more of the polystyrene content. But the ions, for a given ion exchange capacity, the ions are then less distributed or less, fewer of them along the chain. Whereas if you make them short, then the degree of sulfonation is very high to get the same ion exchange capacity. And it's this high degree of sulfonation that gives you very high ionic purity in the ionic channels, very good phase separation, allows you to get much more ions per gram or per material, and that affects the conductivity of these materials. If you take a similar composition, yet you now make a polymer with the grafts further spaced in dimension, going, say, from almost doubling the average distance between grafts, then you give extra room for crystallization of the backbone. And we can see the growth of crystals of the backbone. And that has an effect, again, on water absorption. If you look at systems which are further spaced out with higher crystallinity, this one's about 5% crystallinity, this one's an order of magnitude less. The extra crystallinity restricts the absorption of water, and it means you can go to very high ion exchange capacities, very high ionic densities without dissolution. And that gives you very high conductivity in the very high ion exchange domain. And so again, we're learning how to control the structure and understand it in order to control. Now, for those of you in the fuel cells, you might understand what this means. The point I would like to make in these graft systems, the water contents in terms of waters per sulfonate are actually very small. There's very little liquid water in these, if any. And so in our graft chains, there's about anywhere from 5 to 15 water molecules per sulfonate. Most of that is highly polarized water. This is a holy grail in fuel cell science, trying to get materials that conduct protons with very little, if any, water. And these things do conduct. They conduct really well. These stars are our baseline Nafion. These are based on graft materials. Now, shown another way, if I plot them on a log scale, as you can decrease the relative humidity, you get a decrease in conductivity still. The crosses are for your typical commercial state-of-the-art membrane. These are for the experimental membranes. This slope is actually a good parameter. You want them to be as level as possible, because if you're operating in Vancouver and the humidity is 100%, then you drive to Nevada where it may be 5%, you need to be able to handle that change in humidity. Otherwise, you have to put on lots of ancillaries to control it. A typical hydrocarbon membrane would drop down like this. So these huge improvements. I'm not telling you the whole story, though. And let me tell you what the problem is with this approach. What we learn is this. When you have water around, almost anything can conduct protons. Even systems with low ion exchange capacity can conduct protons because they absorb lots of water, move protons around. The difficulty is when you go to lower relative humidity and they lose the water. And that's where you need systems with high amount of acid, so that when you lose most of the liquid water, you have enough volume concentration of ions to transport proton from ion to ion to ion. So you basically need a solid acid. The difficulty in the systems I've shown is this. When you take materials with extremely high ion exchange capacity, they have a tendency to absorb water when they're in contact with liquid water. They swell like gels. That's not practical. They become gelatinous, very little mechanical properties. And when you go to very low relative humidities, such as in a manufacturing process, you now realize that you're dealing with a material that's largely acid, an acid salt, held together with a little bit of hydrocarbon. And so the mechanical properties are terrible. They're brittle. You can't put any stress on them. Now, in a fuel cell, on the cathode, you've got liquid water. On the anode, they're almost dry. So on one side, you've got a material that might be very highly gelatinous, on the other side, brittle. It's a real problem. And that stress is a large cause for the lack of durability of fuel cells. And so you've got a problem with very high ion exchange capacities. So I'm going to go very quickly and say what we need is we really need much lower ion content to get good mechanical properties, but you need the ions concentrated. That band of ions you see needs to be pure iron and not contaminated with a lot of hydrocarbon. Now, we can demonstrate this using blending technology. It's not sophisticated, but it does the job. What you want to keep is you want to keep your ion channel constant and add a second matrix that goes into the non-ionic to give you mechanical properties and still doesn't interfere with your ionic channel. You can lower an ion exchange capacity simply by reducing the degree of sulfonation, but if you lower the degree of sulfonation, you end up with these isolated ionic groups that don't communicate. So we can blend. And what we're able to do, and a bit better than the others, is that we can control blending materials. So we can control the length of the second component that we want to add to either match in between the sequence length, or we can add the same block length as our fluorous block in the monomer. So I'm going to take these polymers and blend them with fluorous materials. So if you look at a dye block, it looks something like this. These are your ionic channels. If you add more fluorous material, you start to see these larger globules of fluorous, the ionic channels remain intact, a bit more fluorous still, but you can still see these channels being formed. And this is an accumulation of all those micrographs. And basically, I'm pointing out, as you increase the fluorous content, your ionic width becomes similar, but the fluorous materials become larger. But we do see a lot of breakages of these channels, and I'll come to that in a second. In the grafts, on the other hand, which we can add and control the molecular weight of the second component and the sequence length, the grafts start out looking like this, and as we add more and more, this is almost 50% fluorous addition, then there's not much change in morphology. The fluorous domains get a little bigger, but the channels look equally connected. A couple more data slides showing the effect of this. If you look at the effective mobility of the graft blend, of the dye blocks, as a function of ion exchange capacity, as you lower the degree of sulfonation, you get a decrease in proton mobility. That's because the ionic channels are becoming separated. But if you blend, you also get a decrease. And that's because we're getting a breakage of some of the channels. And I'll show this schematically next slide. In the graft materials, if we lower by decreasing the degree of sulfonation, like chemically, you get isolated ionic groups, low mobility. But if you blend, you can keep this ionic connection. And so schematically, it looks like this. In the dye blocks, you have fluorous and you have ionic groups which are connected. If you reduce the degree of sulfonation, then you simply get fewer ionic aggregates and you get a miscommunication that lowers mobility. If you blend, you keep the ionic purity of the channels, but you do have a constraint on the morphology, and we can demonstrate that. Whereas in the graft structures where we have crystalline material, we have hydrocarbon, styrene, and then sulfonated. If you reduce the degree of sulfonation, you get isolated channels in this styrene matrix. But if you blend, it simply goes into the fluorous regions which are connected, and you don't disrupt. And so the bottom line of this is by going into these grafts and getting very high purity channels and then backing off and diluting the ions by blending, you can get actually very good proton conductivities with very limited, with dependence on relative humidity similar to Nafion. And the advantage of the blend systems is this. If you take the pure graft, which are these circles, and you put it into water, liquid water, they swell a huge amount. Whereas if you take these blends and you put them into water, they've got limited swelling on the ballpark of what you see for Nafion. So that's a relatively quick overview. What I've tried to demonstrate is that in order to make advances in some technologies, you have to step back and you have to make model systems, systems which may not be directly applied, but the information you get can give you that leverage for the design of new technological materials. So I've tried to show you how just by changing the ionic groups can change the morphology, how that affects water absorption, how it affects, how you can control that through blending, the importance of ionic purity. And though these are model systems, we actually take them and we work with various companies and we apply this philosophy to much more robust materials. The next 5 years, you're going to see major advances in hydrocarbon membrane technology commercially. And they don't all make use of this, but the principles are not that different. And we make use of this in new types of ion conductors, such as these OH- conductors. And so again, the importance of trying to step back and do some basic research to try to make a leap forward in technological innovations. A bit more information you can find from a review that I wrote with my co-worker Tim Peckham, and also a very nice review by my peers down in the US in macromolecules. The people that did this work, Amy Yang, who's just about to give birth and defend a thesis at the same time. Thomas is an undergrad from Germany. He's now doing graduate work. A lot of the early work done by Ken Shi, and each of these have had some impact in some of this work. And that's to tell me to be quiet. Thanks again for visiting your wonderful university. Look forward when your institute is filled with instruments and people and students. It's great.
Thanks very much, Steve. There's time for questions. Michael?
Very interesting talk. You mentioned in one of your slides that cross-linking is not desirable to improve the mechanical properties between swelling. Is that the reason why that's not the case?
From a manufacturer, there are groups that look at that, and it's a reasonable, logical extension of the work to stop the swelling, but it does have some issues with quality control. So in the back of my mind, I'm always thinking, how do you make 100,000 square meters of these things? And so that's the issue with cross-linking, trying to get uniform cross-linking and getting uniform properties. And cross-linking adds an extra step to cost as well. So you can do that. You can limit swelling. You can get demonstrations and show fuel cell. But I'm not convinced yet that it will be a long-term strategy in commercialization.
Acrylic acid in your structure and then UV cross-linking you get your...
Well, it's possible. Yeah, you can do that. But to do it on mass scale is not as easy to control that level. And again, it just adds another step. It is a possibility. Don't forget, you've got to put the ionomer into the catalyst layer as well, because you need proton conduction in that 10-micron film. And a lot of studies show you need compatibility between that material and your membrane. And so that's another issue. But it shouldn't be ruled out, but it's not I can almost predict what the properties would be. And so we've not gone down that line. Yes, sorry. They're absolutely worse in terms of direct stability against free radical attack. But the companies are interested in these because, and we've got quite a big program looking into this, the question comes from where do the free radicals come from? And if it is indeed gas crossover, like when the engine is being idled or not being used, then the permeability of hydrocarbons is an order of magnitude lower. So even though they may not be stable to direct, maybe not as stable to radicals directly, the overall rate of radical formation may be substantially reduced. That's the philosophy of a lot of people doing this area.
Linda?
Yeah, that's a good question, and it's one that we didn't resolve until a few years ago. But our observations are this. The morphology we get in a dry state is retained as they swell to a certain degree. And then the systems absorb more water than what we see swelling from the scattering of neutrons. So that more water comes in the formation of new channels. So it's almost like the structure's maintained, but then there's another percolation pathways that get formed due to these spurious ions that are not exactly living in this ionic channel. But in terms of the fluorus, unless you go to really, really high water absorptions, 90% and things like that, where things really become different structures, the structures are maintained pretty good, actually.
And with all those ions and those
Yeah, they do. There's a total inversion when you get over a certain threshold of volume. It's no longer good to think about them as water channels. It's more to do with fluorous rods interconnected with water in between these rods. So there is believed to be certain phase morphologies at a very high water content. That's a good point. You know, there's been very little work using fluorescent techniques to examine the structure and dynamics of these. Very little work.
Any reason?
I don't know. I'm trying to think. I mean, people have used almost every single technique you can imagine. But I'd have to think of an experiment, what kind of information we would want and how you would get it. But maybe it's just not had those people address these materials. But I can only think of one example of people doing fluorescent studies on these.
Because the domain is 2 nanometers, 3 nanometers, it simply looks like my cells. And
Yeah.
the electrons have been extremely powerful to characterize my cells.
Oh, well, we should have another talk afterwards. Yeah. That's a good question. Typically, looking at ion exchange capacities above 2 to get the kind of conductivity at low relative when the water's gone. So, but you raised the other question: how important is the acid in the dissolution of platinum? Right, so, and in fact, that's another reason people are looking at hydrocarbons with sulfonic acids rather than these trifluoric acid systems, that fluorus, which gives off fluorine, you know, hydrogen fluoride. And I think it's that's the hydrogen fluoride that's been helping to stabilize platinum particles in the membranes and dissolution. So I'm not sure if it's the acid content itself, the acidity. My understanding it's more like the byproducts. I mean, you do get dissolution of platinum, right? And it forms a platinum band in the material. But if you're doing electrochemistry out of platinum, you basically need an acid group there to do the electrochemistry. You can't avoid that. I can't see how you can avoid that from a practical point of view. Because the acid group's sitting right, it's, you know, a monolayer. It's in the catalyst, it's, you know, a few nanometers thick over the platinum. Highly acidic. pH of that is really, really low. I don't think you can. Yeah, you have to come up with a completely new set of chemistry to solve that.
Then I was going to ask about phosphate groups developing at the end. Can you not add radical quenchers to these membranes to inhibit these radical reactions?
Every single commercial membrane probably got radical.
So
Yeah, they all have.
but you would
And even so, then no, you can't. Commercial systems would not operate without a radical scavenger,
Right.
but they just can't. Yeah, they just don't tell you that. But
And
they.
neither did you.
Pardon me?
Neither did you.
No, I don't want to call it.
So it's not, is it really that critical to inhibit some of this crossover? I mean, I know
Yes,
that
because
simply
even with the scavengers, you cannot cannot capture the radicals fast enough. To date, I mean, there's a huge amount of research going on in that area, and we're very involved in it. So the question is, where do the radicals come from? And that's an even bigger question. Is it peroxide that gets decomposed, or is it a byproduct of the oxygen reduction reaction, or is it hydrogen absorption on the anode? It's a can of worms out there, but it doesn't look like you can stop the radicals with the current technology.
Okay, now, obviously there's probably some crossover just with loss.
Yeah, and you typically want things in the hydrophilic domains to capture radicals, and you know the amount of water that's being produced here is just huge. It gets flushed out. So, and then you've got iron around that decompose. It's a real mess, actually.
So fussy question, in the slopes that you kept pointing out for the grafted and the lamellar versus Naffium, I was a little surprised to see, for example, that the short graft material had about the same slope as the long graft. Long chamber.
Yeah.
Slope. Is that
Yeah.
just because I don't understand the chemistry? Or something?
No, it's when you change one parameter, you get a change in one of the properties that helps, and then a decrease in another property that doesn't help. So it's a competition of things. Yeah. It is. So it has to do with the water content, and in all the situations we measured, there was always I mean, I'm just showing there was just one small domain, right? So it wasn't a complete lamella like a licorice allsort kind of thing. It was, but the preference is that the lamella lie perpendicular to the sheet, no, in plane with the sheet. So if you're making a membrane and you hold it up, the lamella going this way, which is exactly the wrong way you want. It's the only ones we found that didn't have any anisotropy were the disordered structures and the granules, because it's like a neural network. And in my view, that's to me, that's the best view. So there's groups trying to get ordering channels, and that's nice, and you'll publish, but it's not going to be practical, in my view. Yeah, and all you gain is, a factor of 2, 3, 4. If you can get it preferentially aligned in this system. So, the fourth last slide, which one was it?
The one when you compare the equipment exchange for the different membranes. One more? Yeah.
Yes.
So, if you look at the results, Nafion compared to your new system, it's not much better, right? You want it to be a straight line.
Yes.
So,
yeah.
So the conclusion is that you're not improving.
Well, yeah, it's very difficult. I would say, we've got a big database of materials. The best I've seen to date, what we have is similar to. But it's a huge advance from 5 years ago, which they were all down in the bottom here. So by controlling the structures, I would say over the last 3 years, people have been able to move this up. Then there's some limitations. And one of the limitations in hydrocarbons is the sulfonic acid group. Its pKa is not as low as a triflic acid. So when the water contents are really low, which they are around here, you're only looking at 3 or 4 waters per sulfonate. With the sulfonic acid, you're not getting complete dissociation of the proton. So there are people trying to look at making triflic acid derivatives, which I think will push you up in this direction.
So this Nafion is the latest system that was produced by?
Well, there are new versions of the Nafion, and basically it's got to do with decreasing the graft length, and that increases the ion exchange capacity without them dissolving, more crystallinity. Actually, it's very similar, but in different materials.
So what's the state of development of new non-platinum based catalysts for the platinums? The state of the art, there's some very good promising results in the literature to lower platinum or reduce its content. Taking those to the next level of putting them in commercial devices is a bit of a way off. And then the next thing you have to look at is the stability of these. And because now it's such along the road of commercialization where durability is the main issue. You can't. So I would say there's a lot of opportunities, but there is nothing out there yet that is going to revolutionize the process. But a lot of good opportunities. Incredible advances over the last 5 years. I mean, considering platinum has been investigated for 50, the advances we've seen in the last 5 years have been almost a quantum leap. Yeah, you would have thought that bi- and trimetallic catalysts would be good candidates.
Yeah, so you look at ternary compositions, you're looking at core shell, changing the electronic structure, some unknown entities where they have nitrogen and iron that we don't quite know where they are because they've been pyrolyzed, but they work. And so some good research to be had in that area.
Well, if there are no further questions, I'll ask you to join me in thanking Stephen for a very interesting subject.